Histology seminar. Today we're going to talk about nervous tissue.
All right, guys, so let's begin. Before we start, can someone just tell me what's the basic function of a neuron? No one, so we'll just continue. So basic function of the neuron is the acceptance of stimuli, processing of information, spreading impulses, as well as forming functional connections with effectors and receptors. This is probably your previous knowledge, so we don't need to dwell on this much. Another thing about neurons is that it is a morphological, functional, and a trophic unit of the nervous system. So this is just a statement that you have to memorize, nothing much to it. And then we'll look at the components of the neuron. So first of all, we have the nerve fiber, which is made up of axon in the center surrounding the myelin sheath. And then we have the perceptive zone, which is here, which is the dendrite, which receives stimuli from your skin and from other receptors inside your body. Then we have the integration part, which is the thinking part of the neuron. This is the cell body. And then we have the propagation of the electrical component, which is where the electric impulses, they travel through this area. And then we have the effector component, which is the axon terminal. So this connects to the effectors such as glands, muscle, whatever. So again, I will emphasize on this diagram because sometimes in the exam, professors will ask you draw a nerve fiber and you'll be like, what do I draw? So it's just small dot in the center, axon surrounding by myelin sheath. That's the basic structure of a nerve fiber. All right, and then when we talk about the classification of the nerve fibers, we have functional classification, chemical classification, and morphological classification. So when we talk about the functional one, we have the sensory, which are, they detect things like touch, temperature, and pain from outside or inside the body, and they send signals to the brain. And then we have the interneurons. These are the ones which basically connect two neurons together. So we have different types of interneurons. We have the projectory, commissural, and intrinsic. So the difference between them is that projectory, it connects different regions between the brain and the spinal cord. And then we have commissural interneurons. They connect, right and left halves of the brain. And then we have intrinsic. These are the ones which work in a specific area and they don't travel very far. So you learn more about this in anatomy in CNS, but this is just, the gist of it. And then you have the motor neurons. They can be divided into excitatory or inhibitory. And again, excitatory will cause the muscle to be active or contract, and inhibitory will prevent its function. When we talk about the chemical types of the neurons, we have cholinergic, monoaminergic, as well as peptidergic. So when we talk about cholinergic, it's in the name acetylcholine. They release acetylcholine, which is a neurotransmitter, which is important for, movement, memory, as well as learning. When we talk about the monoaminergic, these use chemicals like dopamine, serotonin, or norepinephrine, which influence your mood, alertness, as well as reward. And then the last one, in terms of chemical classification, we have the peptidergic. So what they do is they release peptides, which are just small protein molecules that help in communication. Now, when we talk about the morphological classification, it can be according to the perikaryon shape or the cell body shape, which could be pyramidal shape, fusiform shape, stellate shape. So stellate would be basically the star shaped, and then basket shapes. And then morphological classification according to the type and the shapes of the processes. So we can have heteropolar or isopolar. Now the difference between heteropolar and isopolar is that heteropolar you can distinguish between the neurite as well as the dendrite, but in an isopolar you cannot distinguish between the axon and the dendrite, or the neurite or the dendrite. And then when we talk about the processes, classification according to the processes, they can be apolar, unipolar, pseudo-unipolar, bipolar, multipolar, and anaxon. The next slide will explain this better. So here we can see unipolar. As you can see, unipolar only has one projection, one branch, so that's why they are called unipolar. Then we have pseudo-unipolar. They have one branch which looks like it splits into two. So you can see here, this is the axon, and then this is splitting into two. And then we have bipolar, which have two branches. We have the dendrites here, and then we have the axon here. And then when we talk about multipolar, it has multiple dendrites which receive the information, and oh, sorry, it has multiple dendrites which receive the information and a single axon. And then when we talk about anaxonic, you can see it does not have a definite axon, so that's why it's just called anaxonic. So, guys, does anyone have any questions about the classification of the neurons? No, so we'll move on. Now, when we talk about interneuronal synapse, so synapse is just a functional junction between membranes of two cells. Uh, and it at least requires one neuronal cell. So one neuron cell basically means it just requires one neuron to be there, and another cell can be receptor, effector, or neuroglandular. So when we talk about types of different synapses, we have the interneuronal, neuroreceptor, neuroglandular, and neuroeffector. So neuroreceptor connects a neuron to a receptor, and neuroglandular connects a neuron to a particular gland, and neuron effector could be connection between a neuron and a muscle. So synaptic vesicles, these are just nothing but small bubbles within the synaptic membrane, and they contain different chemicals. These include neurotransmitter, neuromodulator, and neurohormone. So now when we talk about neurotransmitters, these are nothing but chemicals that send messages from one neuron to another. Then, when we talk about the neuromodulator, these are the chemicals that adjust or fine tune the way the neurons respond to the other signals. And then we have the neurohormones, which are the chemicals that act just like hormones. And one important thing to remember about the synaptic membrane, which is the area here, that it has adhesive contacts like macula adherens or punctum adherens. Again, this is the Latin term for these. These are nothing but glue sticky points and adhesive contacts, and they keep the synapse stable during signaling. So they just hold the synapse in its own place. And then we will talk about the transmission of the nerve impulse across the interneuronal chemical synapse. So some of you might already have learned this before, but we'll do this. So the first step is the depolarization of the presynaptic element. Depolarization is just the change in the charge of the synaptic element, which results in release of calcium ions. So here you can see action potential reaches the axon terminal and depolarizes the membrane. So it will depolarize the membrane, and this will cause the calcium ions to move inside the presynaptic element. Remember, this is the presynaptic element and this is the postsynaptic element. And after that, what happens is that these calcium ions, they will cause, the phosphorylation of the synaptic vesicles, which is just, it adds phosphor to these synaptic vesicles. So they will enter here, attached to these synaptic vesicles, cause their phosphorylation, and then this process will cause the vesicles to release out of the presynaptic membrane into this space by the process of exocytosis. So exocytosis is basically when the membrane of the synaptic vesicles will fuse with the presynaptic membrane and release their contents. And then the mediator will bind to the receptors here. So whatever the vesicles are released, they'll bind to the receptors here on the postsynaptic membrane, and then they will just cause the depolarization of the other neuron. So this is how the chain of the transmission takes place. And then what happens to the synaptic vesicles after the depolarization has done? So what might happen is that the spontaneous and transient fusion with the membrane will take place. So basically the synaptic vesicles are present here, and they will just fuse with the membrane. So some of them can be recycled back again. Or what happens is that the presynaptic element, it will pull the membrane of the synaptic vesicles towards itself by the process of endocytosis, so that the vesicles come together and they form an endosome. So this image is really good and it summarizes the entire process. And if you have any questions about this process, please. No, so Ria will take over.
Okay, so this slide talks about the synaptic vesicles and their shapes. There's nothing much to explain particularly in this, but you must know what all shapes are present and what their sizes are along with their mediators. This is very important because they could very easily ask you this information. So I'm just gonna skip to the next slide because this is just a bunch of facts which you kind of have to memorize. Now, there are two positions of the synapses which we can find. There's a presynaptic neuron, which happens before the synapse occurs, and there's a postsynaptic neuron, which happens after the synapse occurs. In the presynaptic neuron, honestly, there's nothing much going on here. There's nothing much to know. But in the postsynaptic neuron, there are like three types of synapses which can happen. There is the exosomatic one, the exodendritic one, and the exoaxonic one. The exoaxonic one is further divided into three parts. It can happen in any of these three parts. The first part is going to be over here between the axon hillock and the initial segment with this part which it takes place on. And in the other part, which it can happen, is like right before it terminates over here, because this is the terminal part. And the third part is where it terminates, like right here. These three are the locations where the postsynaptic neuron can get activated in the exonic one. Apart from that, like just to summarize, there's three types of post synaptic neurons to begin with. Now, let's talk about the neuroglia of the CNS. Neuroglia are just like these special type of cells which are found in our brain. And in the CNS, there's like multiple types, which I'll be going over right now. First type is the astroglial cells. You see here these star-shaped or like stellate-shaped cells. These are called as the astroglial cells, and their main function is just supporting function of the CNS. They help in supporting of the entirety of CNS. And you see these processes which they have. They have got these special receptors which help in like maintaining and checking of the osmotic balance of all the intracellular fluid which is found in the CNS. Their processes, they also cover like two layers of the CNS, of the surfaces, the superficial and the perivascular limiting membranes. These two are really important terms which can be used for astroglia, which you have to remember at any cost. And astroglia are further split into two types. There are the protoplasmic astrocytes and the fibrillary astrocytes. What is important to remember over here is that the protoplasmic astrocytes are only and only found in the gray matter, whereas the fibrillary ones are found in the white matter. This is like the most important thing you need to know about the protoplasmic and the fibrillary astrocytes. I hope that is clear. Now let's talk about the microglial cells. They are basically the scavengers of the CNS, and they have a lot of lysosomes. So as we know, the basic function of a lysosome is to like digest and degrade a cell. So that's why they're called as the scavengers of the CNS, and they are derived from the monocytes. Now, the ependymal cells, they have microvilli and cilia, which help in lining of all the CNS cavities, and they do not possess any basal processes. But there's an exception to this part, and there are these specialized cells called as tanycytes, which fall under the category of ependymal cells. These do have basal processes. That's the exception over here. Like you need to remember that tanycytes are the ependymal cells which have basal processes. Now, the epithelium of the choroid plexus is the choroid plexus. It has many microvilli, and the most important function of it is producing the cerebrospinal fluid. And basically, your brain cannot survive without cerebrospinal fluid. So, choroid plexus is very essential. Does anyone have any questions about the microglia of the CNS so far? All right, I'll move on to the next slide then. There are also another type of neuroglial cells which are found, and they are called as the oligodendroglia. They have, again, further classified into two parts. There's these satellite ones and the interfascicular ones. Satellite ones are only found in the gray matter. This is very important. They are only and only present in the gray matter, and they have like metabolic and trophic influence over the nervous cells. Whereas the interfascicular ones, they are only found in the white matter of the CNS, and they help in formation of the myelin in the CNS. So this part is very important. Like oligodendrocytes, especially the interfascicular ones, they help in formation of the myelin in the CNS. Does anyone know what helps in the formation of myelin in the PNS? All right, I'll get back to that later on then. Uh, there's two type of nerve fibers which are found in the CNS. There are the myelinated ones and the non-myelinated ones. The myelinated ones, they have all different diameters, and the nodes of Ranvier are not present like at regular intervals, are very irregular. Whereas the non-myelinated ones, they are not covered with oligodendroglia, because that just makes sense, right? Because oligodendroglia helps in formation of the myelin, and since they don't have myelin, so they are not surrounded by the oligodendroglial cells. They are components of the neuropil. Now, neuropil is just a space between the neural and glial cells, which contain like a bunch of structures, the non-myelinated fibers being a part of them. Let's talk about the neuroglia of the PNS. there's just two structures over here which you need to remember, two types of cells. There's the Schwann cells and the amphicytes. Now, the Schwann cells, they are like the supporting structures of the PNS, and they help in formation of the myelin, and they also help in phagocytosis. Whereas the amphicytes, they are just like a modified version of a Schwann cell. The nerve fiber of the PNS again has like two types. There's a myelinated one and an unmyelinated one. The only important thing about this diagram is this part over here. Like the myelinated nerve fiber is covered with like reticular and collagen microfibrils. They could very easily ask you this part. Now, there are three sheaths of the peripheral nerve. There's the endoneurium, perineurium, and the epineurium. The endoneurium covers only one single nerve fiber, and it is made up of the loose connective tissue. Now, in this diagram, you see like these white spaces over here. That's just the endoneurium, which is covering only one single nerve fiber. Now, the perineurium, it is like multiple, it has multiple layers, and it surrounds one entire bundle of nerve fibers. So this entire part is the perineurium. As you can see that there's like multiple layers, like a pretty thick layer, and it surrounds the entire nerve fiber. The epineurium is like a structure which surrounds the entire nerve, and it also fills in the spaces between all the nerve fibers, and is made up of the dense connective tissue. This entire part outside over here is going to be the epineurium, and as you can see that it is also coming in the middle over here. It's kind of like joining all the nerve fibers together to form a whole bundle. Yeah, that's about it for my topic. And does anyone have any questions? Okay, then I'll let Katrina take over.
Hi, guys. Okay, can someone just remind me what is a synapse? Can you guys see my screen? Okay, so guys, we just talked about it. So can someone just let me know what a synapse is?
The size of trans.
Sorry? Okay, so guys, because this is like the basics, you guys need to at least understand the basics. So we're just gonna quickly go over it just one little time. Nervous system, guys. Nervous system is what allows us to pretty much feel and also helps us move, okay? And it's composed of neurons. This is a neuron. It's the morphological functional unit of the nervous system. We have it's just composed of multiple neurons and other cells, but this is very important for you guys to know, okay? Quickly, this is cell body of a neuron. Here we have this dendritic processes. Here we have the axon, okay? And the axon is covered by Schwann cells or myelin sheath that are composed of Schwann cells. When we have the information traveling through the neuron, if we did not have this myelin sheath, it would take a very long time for the information to travel. Thanks to the myelin sheath, the information actually will do this. 1, 2, 3. It will travel through what we call the nodes of Ranvier. When you guys say touch a very hot stove, you guys instantly take your hand off that hot stove, right? That's thanks to the neurons. It's thanks to your nervous system, because we have multiple types of neurons. We have sensory neurons and we have motor neurons, interneurons, whatever. When you guys touch a very hot surface, your sensory neurons, sense it, and they communicate with other neurons through synapse. Okay, synapse are how neurons communicate. They communicate to other neurons to then tell your motor neurons, to then tell your muscles to contract, so you guys remove your hand from the very hot surface. So synapses are how neurons communicate. And how they tend to actually do this is through depolarization. I don't know if you guys have done action potential yet. I don't know if you guys are going to do it. But for you, the ones who don't know what depolarization even is, just think of it as a way that it becomes activated, right? Once the neuron, so imagine you guys touch a very, very hot surface, and this neuron is going to become activated, right? And it's going to communicate to the other neuron by releasing the synaptic vesicles. The synaptic vesicles, once they go to the other neuron, they will activate it, causing depolarization. And so we just have this communication of neurons and neurons and neurons and neurons all the way to your central nervous system. And then that's going to communicate to your motor neurons that are communicating to your muscles to tell you to move, to tell you to, do this, contract, do that. So you guys need to understand the basis. Okay, we have the nervous system that's composed of neurons, and we can classify those neurons based on their function. We have neurons that are just for sensory, just for you guys to feel things. You have neurons that are going to help you guys move, that are connected with your muscles. You have interneurons, etc. And then we can also classify neurons based on how they look, just their morphology, okay? Neurons that have one process, multiple process, etc. Just try to remember this basics. Like, this is extremely, extremely important. Right, okay. So now we're going to talk about the degeneration and regeneration in the nervous tissue. So the nervous cells, they are terminally differentiated, okay? That means they are not capable of proliferation. We have sometimes limited production, but overall, if you guys get asked, okay, can nervous cells proliferate? The answer is no, they cannot. That's why once you have some sort of damage to your nervous cells, that's it. A lot of times the remaining nervous cells are just going to have to learn how to take over the functions of the cells that died for whatever reason. That's why it's very important when people, have like a heart attack. It's very important for us to do CPR because we want to carry oxygen to the brain. If the pericardium is damaged, so cell body, if the cell body of the neuron is damaged, the entire neuron, this entire thing, is going to undergo degeneration. And if this neuron is connected to another neuron, right, because they're always communicating, guys. If this neuron is connected to that neuron, then that neuron will also undergo degeneration. If the nerve processes is damaged, so this is like another word for the nerve processes. We're talking about the axon. If somewhere in the axon there's some sort of damage, the entire distal part, so if there's damage here, this entire part, distal to where the damage is, will undergo degeneration. And this is what we call Wallerian degeneration. So again, if there's damage here, this entire thing will degenerate. If there's damage only here or here, the distal part to where the damage occurred will degenerate. Glial cells. Glial cells, they can proliferate. You can have new neuroglia in the adult brain, and this is produced by the neural stem cells and progenitor cells. Peripheral nerves, and here we're going to talk about Wallerian degeneration and regeneration. Okay, yeah. So this is a diagram that is in the lecture that you guys had. I tried to find better diagrams, but unfortunately, like the best diagram is really the one that's in the lecture. And that's good because, now when you guys study this on your own, this image won't scare you because you guys will understand what's going on. So guys, this is a neuron, okay? This is a nerve cell. It's cell body, pericardium, the axon, the nodes around the which is everything in between the myelin sheath, which is where the impulse travels. So once we have depolarization, when the neuron is activated, the impulse needs to get to the other neuron that's here, right? So how does it do that? It has to go 1, 2, 3, 4, boom, boom, release synaptic vesicles that are then going to release, whatever neurotransmitter to activate the other neuron. If we didn't have, again, this is important because professors will ask, why is myelin sheath important? If we didn't have this, then the impulse, instead of traveling like this really fast, it would take its time traveling throughout the entire axon. So again, here we have, right, a neuron. Let's say that there is some primary irritation or a lesion. The lesion is here, guys. Because the lesion is here, what is going to happen? Like we just talked about it. Could someone tell me what's going to happen where is the degeneration going to occur? So the degeneration is going to occur distal to the lesion. As you guys can see, Here we have the myelin sheaths. They're really well like structured, and everything distal to the lesion just seems a mess. And you'll see here lipid bodies, which are a sign of degeneration, and you're going to have fragmentation of the nerve distal to the injury. So this is like fragmentation of the nerve distal to the injury. And another thing that you're going to have is increase in the volume of the pericardion. So there's going to be increase in the volume here, and you're going to have dissolution of the Nissl substances. This is known as chromatolysis or trigolysis, and I remember that this is something that they do ask of you. They might ask you guys, okay, like what happens when there is some lesion on a neuron? And you can say, well, first, if the lesion is in the process, in the axon, we know that there's going to be degeneration to the distal part of that neuron. And that degeneration is, classified by lipid bodies infiltrating the myelin sheath. Second thing that's going to happen, you're going to have an increase in the volume of the pericardion, and you're going to have chromatolysis or tigrolysis. And they're going to be like, what's tigrolysis? Tigrolysis is the dissolution of the Nissl substances. And everything that's degenerated, the Nissl substances, everything that's degenerated are going to be phagocytosed by macrophages. Now, Schwann cells, they can proliferate. So this is actually a good thing, because now we're going to have rebuilding of the area that was degenerated, that was distal to the injury, which is what's happening here. We have proliferation of Schwann cells, and eventually we have rebuilding. We have migration of the nucleus to the peripheral position of the pericardium. And so after the Schwann cells start proliferating, there are two things that can happen. We can have, so now we have a proper, kind of build. So it's not exactly the same as it was before. If you guys can imagine how it is, how a proper neuron would look like, which you guys know because we've been showing them in every slide since the seminar started. So you guys can see it looks a bit different. But there are two things that can happen after we have proliferation as it's being rebuilt. We can have a proper neuron where the function is restored, or we can have a neuron where the function is not restored. And the one that the function is restored, you have like the proper axon, you have remyelinization, you have the Schwann cells, the motor end plate is restored, it's connected to muscle fibers, etc. And everything is working fine. Or we can have one that it's there but it's not functioning. And this is essentially how it would look like. You just have a lot of connective tissue, a lot of scars, and you can see something known as amputation neuroma. But these details, as far as I remember, they might not ask you. They might just say, what's the end goal of regeneration? And you guys will say, well, you can have a neuron where the function is restored and everything is fine, or you can have a neuron that has no function. Okay, so guys, remember when we are classifying neurons, we have motor and sensory, right? So we guys know that the motor neurons are connected to the motor end plate, which is the muscle, because these are the neurons that tell the muscle, hey, I need you to contract, because the information that we're receiving from the sensory neurons is that this person is touching something that's very hot, or that this person is touching something that's causing them pain. So we need you to contract. So the motor neurons are connected to the motor end plate. Sensory neurons, we have what are known as like the sensory nerve endings. What are they connected to? So we can classify what they're connected to according to their function. So you have sensory nerve that are connected to like extra receptors, and these extra receptors are the receptors that accept stimuli from the outside. So they're the ones that are going to tell you what you're smelling, what you're tasting, what you're seeing, what you're hearing. All of these things are information that's processed by your sensory nerves and take that information to your brain. You can have interoreceptors, and they take stimuli from your viscera. So when you have visceral pain, chemoreceptors of aortic and carotid bodies, baroreceptors, these are things that you guys might still be confused as to what they are. Don't remember what topic goes more into detail about this, but just know that you have interoreceptors from your viscera. And then you have proprioceptors. And proprioceptors are when you receive stimuli from your locomotor system. So this is what tells your body the movements that you're making, whether you're in a tense position or just what position and orientation you're in. And proprioceptors are very important, guys, because it's what gives you your posture. It's what allows you to just be able to stand up and not like balance and fall. So this is according to function, right? We can have sensory nerve endings that are connected to extrareceptors, interoceptors, or proprioceptors, and they're receiving all of these informations. Now, not every nerve ending is connected to those receptors. We can classify them according also to just their structure. We can have nerve endings like what we call simple free endings. So they end in the epithelium. They're in the epithelium and that's where they end. And these simple free endings we can find them in the epidermis, oral cavity, respiratory epithelium, etc. We have compound free endings. An example of this is the Golgi tendon organ. This is something that will be very important for you guys in anatomy. In histology, I do believe it was also important. So just the Golgi tendon organ is this organ that's inside your tendons. It's inside your tendons, and basically what it does is anytime there's a lot of tension in your tendon, it activates and tells your muscles, like there's a whole stimuli that occurs, or sorry, a whole processing of information, the Golgi tendon organ, that will then send information to your motor neurons that are going to tell your muscle to contract because there's a lot of tension in your tendon, and this is to prevent your tendon from breaking or, protecting the breaking of the muscle. So the Golgi tendon organ is very important because, it helps you guys with like lesions. And this is something that you guys will see a lot in like athletes, footballers, people that exercise, or even if you're just walking, if you accidentally slip or just are in a position that has a lot of tension in your tendon, what protects you from actually like breaking it or harming yourself is the Golgi tendon organ, because it will activate and it will tell your motor neurons to be like, hey, tell the muscles to contract quickly because this person is about to like hurt herself. And then you guys have the neuromuscular spindle, which you guys already know. So the neuromuscular spindle is inside the muscle, and basically what it does is it has receptors that sense when there's like stretching of the muscle. That's then going to send information to cause it to contract, like a reflex sort of thing. We also have, so we had free nerve endings, or going back. So when we classify the nerve endings, we classify them according to function, which is the extrareceptors, interoceptors, proprioceptors. We classify them according to structure. Structure, we have the simple free endings, which end in the epithelium. We have the compound free ending, which is good. An example is the Golgi tendon organ, and you have the neuromuscular spindle. And now we have encapsulated nerve endings. So these encapsulated nerve endings, you can see them in your slides. I would say the most important ones, the ones that I know, I remember we saw them quite often in our slides, are the last two, which are Meissner's corpuscles and Vater-Patinian corpuscles. But just for you guys to know, we have the Ruffini's ending, and know that the Ruffini's ending occurs in the dermis. We have Krause's ending, which occur in the genital corpuscles. Now, Meissner's corpuscles, they are formed by fibrocytes. They have unmyelinated nerve endings, and they're found in the dermis. So capsule is formed by fibrocytes. They have unmyelinated nerve endings, and they occur in the dermis. So this is an example of Meissner's corpuscle, guys. It takes a while. I know from experience that it takes a while for you guys to be able to differentiate, like once you see a nerve or a neuron from just normal connective tissue. But that's the thing. You guys are just going to have to practice, and then suddenly it's going to be very easy for you guys to differentiate it. But in this image here we have the connective tissue. This is a nerve. It looks different. I don't know how to explain, but it looks different. And this is Meissner's corpuscle. It's found in the dermis. And the function of Meissner's corpuscles, it's there to sense light discriminatory touch or pressure perception. So Meissner's corpuscle is kind of what allows you to sense like this is smooth and something like this is rough. So light discriminatory touch is thanks to your Meissner's corpuscle. You have your vater patinian corpuscles, and these are very complex sensory corpuscles, and these have myelinated nerve fibers. And they're surrounded by Schwann cells. So this is the vater patinian corpuscles are very, very easy to notice, guys, because they look like onion rings, and this is how they look like. Does anyone know how to tell apart a myelinated and unmyelinated nerve fiber in histology, guys? Like when you look at a slide, do you guys know how to, okay, this is a nerve, but how do I know it's myelinated or unmyelinated? Does anyone know? Okay, so we know how the myelin looks like, right? We know that it's these basically white structures. And we know that they are covering the axon. So try to like imagine, or I think we should start with myelin. I mean, you can't really see it here. Obviously, I can't zoom in, but essentially you're gonna have white. You're gonna have white that you're going to be able to see. And this is when you know that there's myelin versus here. If you don't see any white, then that's a sign that it's unmyelinated. But when you see like what, it's very difficult to see in this this angle, but basically, yes, usually when you don't see any sort of white there, then you know it's unmyelinated. And if you were to see like white, then you know it's myelinated, that there's a myelin sheath covering it, covering the axon. Yeah, and oh, and vater patinian, it's there for vibration and pressure also. So this is the reflex arc. Usually, guys, the nervous system, how it occurs is you have your sensory nerves, right? Your neurons that are connected to a bunch of receptors in your body, right? And anytime you guys touch something, sense, smell something, whatever, it's going to take that information to your spinal cord, or to your central nervous system, to your brain. And then from your brain, you have your motor neurons that are just, or effectors that are going to react in the appropriate way. So you can smell something, it's taking that to your brain, and then it's going to cause you to vomit because you don't like the smell of that or whatever. That's usually how it works. It's a whole system, up and down. However, sometimes we have this thing also known as the reflex arc, and that's what allows us to kind of, because this takes a while, right? It can take a very long time. So with reflex arcs, these are instantaneous reactions. And what happens is, imagine, guys, just follow me with this diagram. It seems like there's a lot happening, but it's really simple. You have a hammer, and I hit your patellar tendon. In your muscle, this muscle, you have stretch receptors. Because I hit it, it's going to stretch, right? And the stretch receptor, which is connected to the sensory neuron, is going to carry that information through the sensory neuron to the cell body of the sensory neuron. This is anatomy. You guys don't understand what that is yet, but just know that it's carrying that information to your spinal cord. You have interneurons that are going to connect your sensory neuron and your motor neuron. So the information, so basically it's like this. Something just hit me, or I'm stretching a lot. I'm stretching a lot, and this is going to cause me harm. Hey, pass on the information that I'm stretching a lot. Your interneuron is going to be like, hey, motor neuron, something is happening. There's a lot of stretching happening in that muscle. I need you guys to pull it back. The motor neuron is going to be like, okay, guys, we need to go and pull back that muscle. So now the biceps femoris and quadriceps femoris are now going to pull back. They're going to contract, because we want to get away from the source of pain, which is this thing. And this is known as a reflex arc. So instead of the information going all the way to your brain, which is usually what happens, there are synapses. This is synapses, guys. They're communicating just in the spinal cord. And this is why it happens instantly. This reflex arc is something that is very important, guys. There are people that are asked about this in their anatomy final, and they don't know how to explain what it is. This is really all that it is. It's, you have the stimuli here. your stretch receptors notice that something is happening that's causing pain. Just in the spinal cord, they're going to synapse with the interneurons, which are going to synapse with the motor neurons, and the motor neurons are going to cause the muscles around it to contract. This is a reflex arc. And again, remember the synapse, guys, we're having neuron and neuron communicating. They're releasing, there's depolarization, they're releasing synaptic vesicles. The synaptic vesicles are just going to release, for example, ions that are going to cause depolarization of this neuron, which is then going to cause release of its synaptic vesicles that will cause depolarization of this neuron. They communicate through synapses and through depolarization. So that is really the basis. Please remember that. And that's it. We have a mock micro. Do you guys have any questions about what I just talked about or anything we've done so far? I know the nervous system can be a bit difficult, so like please feel free to ask anything. Okay, if not, this is all for today. Thank you guys for coming. We really appreciate it.
Thank you. Bye.